We are going to go ahead and get started, and I'm going to turn it over to Ying. And
so if we start at 5:15,
yeah,
Hello.
So we're fine. So okay.
Hello, good afternoon, everybody. Thanks for staying for the last section of the day. I know it's a long day, and I hope you guys all stay awake. Can I see everybody here? Good, great. So, okay, so it's my great honor and pleasure to introduce my co-moderator and Professor Yibin Wang from UCLA. And Yibin is a professor of anesthesiology and medicine, and he's also the vice chair for research in the Department of Anesthesiology. So his title is Cardiomyocyte Maturation in Development and Stem Cell Biology. Here you go, Yibin.
Thank you, Ian. And I also wanted to thank Joe, GQ, and Jay for inviting me. This is really not the area that our lab has been focusing on in terms of cardiac regeneration, but we've been very interested in some basic biology that potentially can be applicable to the stem cell biology as well as regenerative medicine. So I really venture out of my comfort zone, so hopefully we'll get some input from you. And this work was really spearheaded by a graduate student, Josh, and initiated originally with some of the discovery by Chen Gao and a lot of support from informatic aspect by Christopher Arm. So our lab has been very much interested in kind of both signaling, but also transcriptome regulation in cardiac development and a disease. And what we recognize from our very kind of a naive observation is that when you look at cardiomyocyte differentiation and lineage determination, you can always see that in the fetal stage, you can always identify many of the molecular signatures that defined by transcription factors and signaling molecules, as we listed here, that really determine the fate of those cells, and to the point that you can actually use the combination of those transcription factors, turn fibroblasts into cardiomyocytes. But when we move down to a sort of determined or differentiated cell types, like fetal cardiomyocytes towards adult cardiomyocytes, and suddenly we realize that all the features start to focus on the function rather than the regulatory kind of underlying drivers. And indeed, if we look at from fetal cardiomyocytes to adult cardiomyocytes, there are tremendous change in many aspects, from structure point of view, from functional point of view, metabolic point of view. Yet the underlying mechanism appears to be poorly understood and really understudied compared to other aspects of the cardiac development. So again, from a very naive point of view, and we thought that this could be a very interesting gap in our knowledge that we can tackle. And as it turns out, this is also a major limitation in our current stem cell biology or stem cell either therapy or modeling, where the either iPS cell-derived cardiomyocytes, or in fact many other sources of derived cardiomyocytes, they nevertheless reach this sort of cardiomyocyte feature, but actually in an immature form. So myocyte maturation is actually a relimiting step in terms of stem cell biology as well as its application. And so a number of years ago, Marlon Torma, who is actually a pediatric fellow, came to our lab and started to do a Ph.D. in my lab. And she was very much interested in this a window of maturation where in a mouse it would be about 7 days. And so we designed a study to use transcription regulation as a driver to actually identify what's the underlying forces that actually help the maturation process. It was a very naive thought that if we know all the genes changed, we may be able to identify the underlying sort of drivers or transcription factors and so forth. So you can see that by principal component analysis, many genes changed. And the biggest difference is, of course, is the time. It's from P0, P3, P7. And if you look at left versus the right ventricle, their difference is actually pretty modest compared to this developmental time point driver. So clearly there's a change of gene expression, but we will look at their kind of a mechanism or impact. It's no surprising that they affects many of the cellular processes involving metabolism, cell proliferation, and so forth. And so we were trying to look at what is the underlying regulatory sort of a scheme or network that really drive this process. And lo and behold, that we didn't. We failed to look at, or identify either master transcription factors or list of or number of transcription factors that actually help us to explain that dramatic gene regulation. However, we do find, at least in the geo-analysis, that some of the top hit sort of regulatory genes actually involving mRNA splicing. And so we went back and looked at, it turns out, much of our genes are indeed alternatively spliced. In fact, there was an estimate about 80% of the genes expressed by mammalian genome are alternatively spliced. And this alternatively spliced transcriptome clearly can generate different proteins and also help to not only enrich the complexity of the transcriptome, but also indeed fine-tune the cellular function at multiple levels. And so we look at many of the known splicing variants that we know that will be actually changed during development. And indeed, if you find many of those exons that are alternatively spliced during this maturation period between neonate to adults, and interesting enough, many of those same exons or same alternative splicing events will be reversed in the failing heart. So we feel that there is perhaps a what we call the neonatal reprogramming or fetal-like reprogramming of RNA splicing also happening in the diseased heart that make us more interested to look at what could be the underlying mechanism. And among many of the RNA splicing regulators, we looked throughout both the neonatal and the adult and as well as the diseased heart. We found one of them is very interesting, that is the RBFOX1. It's turned out to be more than 30-fold increase during this critical period of time of 7 days. And RBFOX belong to this RBFOX1. It belong to the RBFOX family, which has a 1, 2, 3 isoforms, and the two main isoforms of the RBFOX1 expressing both the muscle and the brain. And much of the literature actually focus on the brain isoform. And what we learned is that RBFOX1 in the blue here is actually hugely increased from undetectable to very high level, and while the RBFOX2 continued to decrease throughout the different maturation and the aging. And we can recapitulate some of that effects in the human ESL-derived cardiomyocytes during their differentiation, but you have to compare these two levels of expression. Notice that even in the best maturation state of those human ESL-derived cardiomyocytes, the RBFOX1 expression is much lower than the adult hearts. And so we did RNA splicing exome profiling and recognized that in the RBFOX, all the mRNA splicing events that we observe in this fetal to adult heart maturation process, about 21% of them actually contributed by RBFOX1. This was done by both gain and loss of function studies. So it's a very significant, but clearly not the only one. There are many other factors probably involved, but at least RBFOX1 play a very important role. So we ask a very simple question. Is the RBFOX1 really matters in terms of cardiomyocyte maturation? And then we use this neonatal heart myocytes in our in vitro system to actually test that by expressing the RBFOX1 specifically in the neonatal myocytes. And we see that indeed, you see the calcium handling proteins are much higher. And if you look at the alpha-beta ratio is also changed, increased. And those are the signs of maturation. And we also see increase of metabolic gene expression. And if you look at this, just in the serum-free media, you can see that they actually promote sarcomere organization. And Dr. Van Treska actually reminded us that when we look at those cells, the nuclear is also condensed as a sign of maturation, which was also observed in the in vivo situation. And so Josh actually wanted to look at what is the real functional impact of those RBFOX1 manipulation-induced potential maturation. And so we look at the cells that are implanted on those sort of a matrix that allow us to look at deformation of the matrix, allow us to actually have a direct measurement of the contractility as well as the calcium signal. And this is probably not a very impressive movie because if you look at their contractility under stimulation, was very weak with the LAG-Z treated cells. I hope it's running, yes. And then in the RBFOX1 expression cells, their contractility are a lot more obvious, and I'll show you the quantification later on. I'll have to skip. So you look at the contractility assay. They are detailed quickly after stimulation over time, yet the RBFOX1 expression cells actually can continue for quite some time. And you can see that if you look at it from the single field, you can pick every cell or different cells, they are synchronized. And if you look at the calcium signaling that reflects in the contractility as well, is that those cells, the immature neonatal myocytes actually do not pace well, especially on the high frequency 2 Hertz. But if you look at the RBFOX1 cells, they are much more robust in terms of response to calcium cycling. So this is probably a nicer way to look at in terms of the calcium robustness of the calcium cycling as well as their synchronized events. Okay, so it appears that RBFOX1 expression can promote better function in neonatal myocytes. And this is again the quantification of several cells and in the same in the same field. So, but we know that one of the best kind of or better utilization of this concept would be in the human iPS cells. So we knew that iPS cell-derived cardiomyocytes can be used for many purposes, both for cell therapy but also disease modeling, but that require iPS cell to adapt more mature myocyte phenotype. And so we use the human iPS cell-derived cardiomyocytes. This is initially provided by our colleagues from UCSF and Deepak's lab, and now we actually get regular kind of a shipment from Joe Wu's lab as well. And we see that by expressing RBFOX1, you can drive down the fetal gene, but also you can increase this cardiac or mature cardiac genes. And you can see the beating rate is, or beating rate, simultaneous beating rate is a lot more improved. But if we look at the structure, they indeed have better sarcomere organization and as well as contractility, just like we showed earlier with the imaging analysis as well. And so to test this, what we are trying to do is actually to use that manipulation molecularly, but also combine with engineering, material engineering to actually use this printed platform where the strain can be, or the physical constraint can be actually put on these cells, and they can be printed to single cell length. And this is actually the earlier kind of test we did, and that the underlying fluorescence signal actually can be directly measured for the cardiomyocyte contractility. And you can imagine that if we have this as in the dish of the cells that actually are working with especially patient-specific iPS cells, we thought it will be a better way to look into both the heterogeneity of those cells, but also the functional aspect of the cells. So I just rushed through kind of a simple concept where we believe that more than just the transcription regulation, but also post-transcriptional regulation by RBFOX1 may contribute to the maturation process. This is certainly not the only one. We believe that there are many other probably underlying mechanisms involved in the RNA splicing or RNA processing events, and we're hoping that adding those molecular insight may help to make the cells more mature and more adaptable to both therapeutic aspect as well as disease modeling. I already indicated the major heroes in this study, the Josh and also Chen Gao, and with the contribution from Martin's earlier data and also Christopher's help with the informatics. This engineering part was actually with the D. Carlos lab from UCLA, and cellular physiology and the molecular biology coming from other collaborators. With that, thank you so much.
We have time. Jared?
Yibin, a couple of quick questions about your cells with the calcium signals that were being faced. One is in the Laxi cells, I noticed that the nucleoplasm seemed to show a calcium signal. It seemed to show a phasic increase and decrease in fluorescence with the cytoplasm. Did you notice that? And if you did, what do you think?
We did not, I did not pay attention on that. I'm sorry.
And
we actually measured the whole cell calcium, and this image was not taken under the confocal, so.
So you're saying maybe there,
Maybe
usually in those cells, there's not a lot of cytoplasm on top of the nucleus.
But it's interesting.
And in the RB-FOX1 treated cells, one of your myocytes was the calcium signal would go one way during one cycle and then the other way during the next cycle, and that happened during the whole loop. Did you notice that?
Yeah, I noticed that. That is actually the one with particularly long shape of the cell. So you can see there's kind of a calcium. Yeah, there's a. We don't know how that could be explained, but clearly there are many perhaps intermediate maturation process going on. So.
Very nice talk, Yibin. So I'm wondering whether RB-FOX1 promote ventricular specification versus atrial cell. How about MSE2V2A ratio?
That's a very good question. We actually never look at those two. Yeah, I think we don't know the expression in atria. I'm sorry. Yeah, we should have a look at it. But it's definitely in the ventricle.